I'm Christina again. Nice to be here. I just want to remind everybody that if you have any questions at all, please interrupt me. I want you to understand this, and it is sort of complex, so if I don't do a good job of explaining, please let me know. Thank you, Mike. So the outline for today, talk a little bit about our preclinical data, which is kind of combined with our gene-targeted therapies data. I'm going to touch a little bit on international research updates and then areas for future research. So the goal of this talk today is to sort of update you on our recent work. So these are projects that we've been working on in the last year or two. We have years of other data for mouse work and for our iPSC and neuron work. I'm not going to show those slides today. If you have questions about them, I can talk about them for hours, and I also have my computer, so if anybody wants to see figures or any of the background data that led to what I'm going to talk about today, I'm more than happy to show that. I do have a couple slides just of background that will kind of lead us and segue into the gene therapies data. So just to orient everybody, this is the gene DARS2. So as Dr. Fine referred to, these blue squares here are the exons, so those are the protein coding regions. And so there are 17 in DARS2, and then these areas in between, these are the introns. And so this is a figure from 2014 from the Amsterdam group, and this is showing, at the time, all of the patient mutations that were identified within DARS2. And then the colors represent the different types of mutations. And so here in intron 2, right ahead of exon 3, is sort of this hot spot that Dr. Fine was referring to, where that frame shift mutation is. And that frame shift mutation leads to a skipping of this, and that means that no functional protein is made, and that's not good. And as you can see in the numbers in the parentheses, that refers to the number of cases they have. And so those numbers there's that one mutation, there's 87 records of that. And so you can see that it's just sort of a very prevalent, a very hot spot for those mutations. These blue mutations, these refer to missense mutations, so that's where there's a change in the letter of the nucleotide, and that leads to a change in the amino acid that's coded there. So, that can cause either a change in the protein shape or the way the protein functions, and so that's just something to keep in mind. Next slide. And so one of the really complicated things about genes, and one of the things that account for or explain some of the complexities in humans, is that one gene can produce a lot of different proteins. And so they do that by sort of rearranging these exons and introns. And so DARS2, the gene, can produce 13 different transcripts. So in theory, it can produce different types of proteins or different shapes of proteins. We don't really understand DARS2 well enough to know what those proteins are like, but we know that there are 13 different transcripts. And so the red transcripts up here produce proteins, in theory, that everybody makes and that are normal, and that's fine. This yellow is the full length, so all of these vertical lines are the exons like I showed in the last slide. And so this is kind of what we consider the normal, the full length. Those red ones produce a protein that we are okay with. And then these blue ones are transcripts that are naturally made, but the body doesn't like them, and so they get decayed and degraded. And so one thing that I want to point out is that all the ones that get degraded are missing this exon 3. So you can see in this full length, there's the third exon and all the exons, and all these lack the third exon, the third coding region. And that just means that if there's no exon 3, there's no protein. So these are sort of garbage. And then we need exon 3 in order for there to be protein. And so this sort of forms the basis of a lot of what we're working with the gene therapy, and especially with the ASO therapy. So go ahead on the next slide. So this is a schematic and a gel blot that we use in the lab to sort of determine if that exon 3 is there. And so on the top in the blue is exons 1, 2, 3, 4, and then I have a dotted line all the way to 17, so just assume that those are all there. The red blotch indicates that mutation that a lot of patients have. And then this is something that we do in the lab. So we develop these primers that can stick to exon 2 and can stick to exon 4. And if they can stick to exon 2 and exon 4, we get two products. We either get one product that's 2, 3, 4, or we get one product that's just 2, 4. And then if we run this on a blot, we can see that a control iPSC, so cells that we get from a healthy person, produces a lot. So this blob is bigger of 2, 3, 4, and a little bit of 2, 4. So that's normal. That's just the way it is. But when we run an LBSL patient, we see that this blot is a little bit smaller compared to control, and this blot is a lot bigger compared to control. And that's what we're trying to fix. Next slide. And so the two ways that we're going to try to do this is through AAV9 therapy and antisense oligonucleotide therapy. So this is just a little introduction slide to what's next. Go ahead. Thank you. So actually, go back one, Mike. Thank you. Sorry. So with the AAV9, so the way this works, the AAV9 is a virus. It's an engineered virus that has been used for several years now to deliver a gene. And so we can actually design this virus to contain DNA, and that DNA is the DARS2 full length, and that DNA can be delivered, can enter the nucleus, and can affect transcription. So we can put in a full-length DARS2 that's going to go in and alter transcription to then hopefully produce full-length protein. And then I'll talk about the ASO when we get back. So now you can go ahead. And so what we did is we partnered with some colleagues at the University of Maryland. So this is Dr. Walczek, Dr. Janowski, and Dr. Lang is the, he's a postdoc in the lab who I think is now a research scientist and did a lot of work in designing the plasmid that we used. And Adam is a technician, was a technician in the lab. He just left to go to an MD PhD program in California. And so Adam is the one on our side that did all the work. And the gentleman at University of Maryland designed this plasmid. They gave it to us, and then we tested it in the lab. And so what Adam did is he used this virus, and he gave it to our stem cells, and then he also gave it to some neural progenitor cells. So these are cells that are early neurons, early brain cells. And then he also allowed those early neurons to mature into full neurons, and then we tested different outputs to see just how these cells respond to this AAV9. And then we have a couple different endpoints, and I'm going to talk about all of these, and some of these are sort of long-term endpoints that we're hoping to test in the future. Next slide. And so this is a schematic of gene expression. So this is sort of like that blot that I showed in the earlier slide where we're just looking at how much exon 3 is compared to other exons. And so maybe my animations are a little bit screwy here, but go ahead and just, yep, so you can stop there. And so we have two different patient lines here. So what you can see is that I have three different exons of exon 3, exon 6, and exon 17. So I did this to represent a few different things. So if we have exon 17, I'm just assuming that's a full-length protein. So that we can say this is the amount of full-length protein that some of these cells have. And I've normalized this to 1 here. Exon 6 is an exon, if you remember from that transcript slide, it's just an exon that seems to be represented most frequently. So of all the different transcripts are there, some lack exons further down, but most of them have exon 6. So this is kind of the max number of transcripts that are present. And this is exon 3, so this is the one that is most frequently missing. So all we want to take away here is just the ratio. So we know that compared to other exons, LBSL patients don't have a lot of exon 3, and that's not good. So when we treat with AAV9, go ahead and click, what we see is that we increase the expression of all of the exons, and that's good. So we're getting transcripts now that are full length and that have exon 3. And so one thing I want to comment on is that this gene expression, so this is a fold change on the y-axis here. So that's a 250 fold change. That's huge. That's more than we ever want to do, but this is a proof of principle that if we treat these cells with this gene therapy that's delivering DARS2, we can increase the amount of DARS2 that's actually in the cells. So this is a good proof of principle for that. Next slide. So the other thing that we wanted to do is we wanted to look at if this affects the way the cells grow. And so we have data from a few years ago, and I can pull that up on my computer if anyone wants to see it, that the LBSL cells don't develop as the same level of complexity that a control cell does. So neurons need to have processes, they need to have long axons, and they need to communicate with other cells. And what we see is that while control cells develop a lot of these dendrites and processes, the LBSLs don't have the same degree of complexity. So usually their neurites are smaller, they have fewer branch points. And so what we wanted to do is to treat these cells and then look at those branch points and look at those processes to see if we can improve it. And so what we have on this graph here is day 1, day 5, and day 9 of treatment with AAV9. And then we just have one LBSL cell line here, no dose. So this pink dotted line is sort of how these cells grow over time. and then a low dose and a high dose of the AAV. And you can see this low dose, there's really no effect, but when we do the high dose of AAV, we get an increase in the area. So this is sort of looking at the axons and the growth of these cells over time. And so just click once more. So here I'm showing you, this is just a blow up of the day 1 and the day 9 from this high dose. So, I'm sorry, that's the no dose and the high dose. So comparing day 9 for no dose, this is sort of what the cells look like. And then I think you can appreciate here with the day 9 and the high dose that the area is just much more full of the cell processes, the axons, and there's a lot more interaction between them. And it's almost too much to actually quantitate because a computer program would have a hard time differentiating what's an axon from a cell and sort of what's the background. But I think you can see that there's a difference here, and that's what's quantified in that graph. So this is good because we can change gene expression, and we can also change sort of the functioning of the cells, and that's based on the assumption that whatever these cells are doing in the dish, the communication is increased and that sort of thing. So the function of these cells is in theory improved. Next slide. And then we look at seahorse. So this is mitochondrial function. This is, again, just 2 lines, and we're comparing them sort of to themselves. And so this is an assay that we run in the lab quite often, where we look at oxygen consumption of cells. So this is measuring the mitochondrial function. And so this is sort of baseline. So we just understand that these cells have this level of mitochondrial function at baseline, and then we can treat the cells with different drugs to sort of manipulate their functioning. And so this drops down function, this knocks out certain complexes of the respiratory chain, and then we push the cells to their max capacity, which is what FCCP does, and then we sort of knock it down again. So this is not a perfect profile of what cells should look like in the seahorse, but this is what the LBSL cells look like, and we understand that, of course, these cells have some mitochondrial dysfunction. But if we treat these cells with the low dose of the AAV9, go ahead, we can see that the blue compared to the dotted blue and the red compared to the dotted red, we see that we can increase baseline respiration, we can increase max capacity, and we can increase sort of their the difference between that max capacity and that baseline. So that just means that these cells have more energy reserve than they did initially, and that's important. So this is just another indication that these cells are improving both in their gene expression, in their function, and then their mitochondrial health. Go ahead. So the last step is to test this in vivo, and this is just a summary slide of mice that have a DARS2 deletion in certain neurons of the brain. This is a paper that we published in 2020, and what I want to show you here is that these animals, over time, though they develop normally and they look very close to controls initially, as they age, they have a progressive loss of neurons. And this is reflected by a change in behavior, and it's reflected by changes in MRI. So these animals actually lose neurons within their cortex, and you can see that here, just that difference between control and mutant, and you can see it in the MRI, that white area is just CSF, so that's lost tissue. And then you can see it very nicely in this picture here. And so what we wanted to do is we wanted to see if we can deliver the AAV9 to these mice, and if we can halt this degradation, this loss of tissue. And so click once more, I think I have an arrow. So what we've done now is we've taken about 10 mice, and we have injected them with the AAV9. And so we take a needle and we basically inject into the brain so that it gets delivered into this ventricle. And if we deliver into the ventricle, the idea is that the AAV9 is going to sort of spread into the brain tissue. It's going to integrate into those cells, and it's going to change gene expression. And so what we're hoping to see is that by the time these animals are 24, 26 weeks old, that we don't have that increase in behavior. And hopefully if we don't have that increase in behavior, we're actually preserving some of their brain area. And this is studies that are ongoing now. We have a few animals that are making their way through, but it's still a little bit too early to share that data. So we're crossing our fingers on that. Okay. Any questions so far? AAV9. Go ahead.
Sorry, I probably should have asked this earlier, but can you talk briefly about the DARS2 makes these proteins, and what's the role of those proteins within the cell?
That is an excellent question. And so I don't have a slide on that, but I think I can go back to, you had that in yours. Can you go to, oh boy.
You want me to just...
Go up.
This isn't working how I work PowerPoint.
There's a picture, like a cartoon of mitochondria. That one. Can you want to make that one bigger? Okay, well, we can work with this. This is fine. And so DARS2 in this figure is sort of this gray and pink swirly thing. So that's basically a structure, a 3D reconstruction of what it looks like. And DARS2 is a dimer, so it's just two proteins that are sort of stuck together to function. And it's shaped strangely so that it can hold an amino acid and a tRNA. And so in basic biology, when proteins are made, so there's DNA gets translated into RNA, and then mature RNA gets translated into protein. And so in RNA, there's base pairs, and then each bundle of 3 base pairs is a codon. And each codon translates into an amino acid sequence. And so these 3, three, I usually have a figure for this, and I should have brought the thing. So 3 base pairs is a codon, and then the codon codes for a tRNA. So a tRNA brings a specific amino acid. An amino acid gets built into this polypeptide chain, which eventually becomes a protein. And so it's really interesting because each of these 3 codes for a different amino acid, and each different amino acid requires a different tRNA. And so a tRNA is bound to an amino acid, and then those two become a bundle through a tRNA synthetase. And so DARS2 is a very specific tRNA synthetase that only feeds aspartic acid into that growing polypeptide chain. And so there's 20 different amino acids, so serine is a different one. That's a different tRNA synthetase. And so there's 20 different tRNA synthetases that function to create this growing polypeptide chain for protein. It's kind of I can send you like a schematic of this that makes it a little bit easier to understand, but it's a very specific enzyme that only feeds an amino acid to a specific tRNA so that tRNA can go do its job. And
okay.
Within the mitochondria, I wasn't going to get that far, but
okay.
What's interesting is that, there's 20 different amino acids, so there's 20 different tRNAs, so there's 20 different tRNA synthetases within the mitochondria, but each one of these different tRNA synthetases leads to a different disease. So DARS2 is LBSL, HARS2 is something else, MARS2 is something else, and so they're very unique in their function because they serve different amino acids, but they all sort of lead to a very different presentation when they're mutated. Was that at all clear?
It was slightly above my level of understanding, but I appreciate that. But my question was, they go to the mitochondria, and the mitochondria are very important for cell
energy production.
Energy production, and okay.
Yeah, so the mitochondria have their own system for making protein, and they only make 13 proteins, and those 13 proteins feed this respiratory chain complex, which is what generates the energy. And so, it's a huge job because there's a lot of mitochondria, and these respiratory chain complex components are very important. But, there's only 13 proteins that are made. So,
but the proteins themselves are incredibly complex.
They are.
Figures of and of
They
themselves.
are. For sure. It's just, they're necessary for mitochondrial function in total. And mitochondrial functioning in these patient cells is disrupted because we can see that, and your kids know that. But if we look at translation closely, if we look at the job of actually making these proteins, where DARS2 puts these two things together, and then where these tRNAs come and actually feed this polypeptide chain, it looks like that is kind of working normally. So somehow aspartic acid is making it into into these polypeptide chains, and these proteins do get made, and they're present in the respiratory chain complex. So, from what we understand, it looks like that main role of DARS2 is still there and is still working adequately. But, regardless, there is mitochondrial dysfunction, and, there are symptoms in the kids. So it's something. So I was at a tRNA synthetase meeting 2 weeks ago in Canada, and, there's 100 people there trying to figure this kind of stuff out and understand, what else perhaps these enzymes are doing that leads to these different diseases. So they're really complex enzymes that do a lot of things more than just this in the mitochondria and in the cell.
All right, thank you.
Yes, go ahead.
So it probably is a little bit a technical question, but in the experience that we had looking at the transcript for the intronic mutation, we found that there's a certain, you also showed there's a certain percentage of full length. So like a leaky, it's like a leaky mutation.
Right.
So I was wondering if, giving antioxidants, you could in some way change the situation and increase the full length just with the antioxidants. So that would be interesting to test, at least in the system that you have. Which is the amount of full length, or if you have a certain amount of increase of full length, treating, the
Right.
system with antioxidants. This was the idea I had, probably because riboflavin or something can in some way
Right.
improve something, because one of these patients that has stabilized now for 10 years with riboflavin has this this mutation in exon 3. This was the...
Yeah, so we tried for a really long time to give the animals a NAC. We gave them a potent antioxidant, and we had it delivered by a nanoparticle system so that it was targeted delivery to cells in the brain. And I treated these animals for close to 9 months, and there was not a single change in the trajectory of the behavioral change or in the neuronal loss in the tissue. And so I never actually did any gene expression analysis on that tissue. But it's something that we could easily do in the cells. So we could do it to the iPSCs and to the
NAC
neurons and see how it goes.
Yeah, NAC is one of them, but the antioxidant systems are different. Perhaps
Right.
NAC is not only sufficient, and then another question, but going back to the natural history and 6 minutes walk. Excuse me. Because,
No, that's fine.
did you test in 6 minutes walk the delta between the first minute and the 6 minute? That could be a way to detect any fatigability. That could be an adding information for performing the 6 minutes walk.
That's absolutely right. We talked about doing it, like our research physical therapists, like we've done it for at Kennedy for other leukodystrophies that we study, and absolutely, we could be easy to try that. Yep, thanks. Good suggestion.
Anything else? Okay. Next, yep, there we go. Okay, so now I'll talk about the antisense oligonucleotide therapy. So this is work that Manu presented yesterday for the scientific portion of the conference. And this is really neat technology that is getting used sort of more and more widely. So these are synthetic sequences of RNA, and they're about 20 nucleotides in length, and the body handles them fairly well. So this is sort of like a COVID vaccine was an mRNA vaccine. And so your body takes that mRNA and then it just, reads it and it turns it into protein and the mRNA gets degraded. So, it's a sort of play off of a natural process in the body. And so, it's relatively well tolerated. And so Manu has made this figure. So this is a review paper that he wrote last fall and published in a journal. And this is a really nice figure showing the different mechanisms that ASOs can take. And so this is a system that's widely applicable to several different diseases depending on, how these mutations affect a cell. And so, here, everything is sort of summarized above, and then the specific mechanism that we're interested for LBSL is this splice enhancer and splice silencer. And so again, we're targeting that intron 2 region of the gene because it's a splice site mutation. So within that region of the intron, so the intron is really large, and within that region of the intron, there are different there are different naturally occurring sequences that act as sites that enable the splicing machinery to either perform better or perform worse. And so this is just, normal. This is present in every single intron when a splice occurs. And so in this area of intron 2, there's actually a very active splice silencer. And so that makes it more difficult for the splicing machinery to act normally to put exon 2 together to exon 3. And so what we did is we designed an ASO that covers up that splice silencer. So that makes it harder for that splice silencer to work. And in theory, what that should do is it should make exon 2 and exon 3 come together better. And so that's sort of what we're working with here. And this is a mechanism that's used, I think that's the same one for SMA, is that right? Go ahead. Yeah.
Would this only be effective if you have the leaky splice at that site? Like if you don't have this mutation, would it then not be an effective therapy for?
Correct. So that's a great question. And so, 95% of the patients have this mutation, and so it's kind of an avenue that makes it worthwhile for 95%. But it doesn't work for if a patient has 2 missense mutations or a splice site and missense mutation somewhere else. It doesn't work. The nice thing about this is not, if you have an intron 2 mutation, you don't have to have the same intron 2 mutation. So in that schematic early on, there are 87 people reported to have that single one, but then there were probably 60 others that had different mutations within the same area. So it's not intron 2 specific mutation, intron 2 mutation specific, but it does have to be something in that area to work for that patient.
I understand right, AAV9 could be effective for everybody, and this is a much more targeted therapy for.
That is correct. Yep. Nick.
As a target, you have, for this intronic mutation, as a target, you have a splicing enhancer, or a sequence, or you have or it's different from. I suppose that you have designed the ASO for a splicing enhancer sequence.
So, so we designed 15 different mutations and 15 different ASOs and walked them down that intron 2.
Because the variants in intron 3 are quite different. So
correct, right. Yeah. So, and it doesn't need to be, so we talked to some people that, design ASOs for a living, and if you want to affect the behavior of a silencer or an enhancer, it doesn't need to be, right on top of it. It can be somewhere else, sort of in the vicinity, and still have an effect on it. And so basically what we did is we just took these 15, we walked them down the intron, and we tested them all in gene expression to see which one reliably increased gene expression the best. So the idea is that it's covering up that
to design
silencer.
different ASOs for different sequences. Yeah.
Right. Yep.
Related to different mutations in intronic in 3.
Good. Okay, next slide. Oh, yep. And so I just wanted to point out that this is not a new idea. So this is a paper from the Amsterdam group in 2012, where Dr. van der Knap's group designed an ASO to sort of accomplish the same thing. And this ASO was a little bit different. So we're targeting these splice silencers and splice enhancers, and they had an ASO. Can you go back one, Mike? So they designed an ASO that has this non-complementary tail. And so what that does is it just makes sort of that splice area a little bit more visible. And so that's kind of a similar idea. It's a different sequence. It's acting in the same area to do the same thing. So that's just a different style. And that's what they did. You can go back to that slide. And this is kind of similar to the blots that we were showing. So they generated these mutants in a cell type that had that intron 2 mutation. And what they're showing here is that they're just, with the treatment of this oligonucleotide, they're increasing the larger, the blob that's associated with a larger fragment, just meaning that exon is now included. So it's the same idea. I just want to, talk about that this is something that's been thought about for quite a while and that others have tried. Okay, so here's a picture of Manu. He's wearing the same jacket. That's convenient. And so he has done a lot of this work. It's been a lot of work, and he's done a really good job on it. And he talked about it yesterday, so I'll spare him today from talking about it. But essentially, it's very similar to what I talked about with the AAV9, where we have this ASO. We treat iPSCs, so these are the stem cells that we generated from the patients, and then we drive these into neural progenitor cells, into neurons, and he's done a lot of experiments testing treatment of the ASOs at different stages, so treating them as iPSCs, treating them as neural progenitors, and treating them as neurons. And then, again, some of the similar endpoints that we talked about with the AAV9. Next slide. And so he has some really nice data. So this is neural progenitor cells, so these are the early neurons, and then quite a few different bars here. So these are data from 3 different patient lines. This is a control, so this is iPSCs that we generated from just a healthy child. And then this is the LBSL cells gene expression. So this is looking at inclusion of exon 3. So this is, again, looking at that exon 3, that lowered exon. So this is without treatment. This is one LBSL patient. This is a second one, and this is a third. And you can just see how decreased that is compared to that white bar. And then if we treat with 3 different ASOs, so this is, or 2 different ASOs, this is 1 ASO, 2 ASOs, and then we did a cocktail, so we combined the 2 together, treated them at the same time, and then at different doses. So a low dose, and then the hatched bars are a high dose. And what you can see is that with each of these ASOs and then the cocktail of both of them, compared to the gray bars, we're increasing the gene expressions. We're increasing the number of transcripts that have that exon 3 in there. And that's again, the whole goal. And then for the most part, we also see that there's a dose response. So the higher the dose, the higher that transcript count is. And then there is a little bit of variability in the patients. You can see this patient responded so that they're up to now control levels. And so that's just, good to note that we do see variability in the patients, and we're not really sure, how close to control that level needs to be to get a functional improvement. But this is all things that we're working on. Next slide. So this is looking at lactate. Part of the disease, the last L is lactate elevation. And so that is something that is usually detected on MR spectroscopy in patient brains, and this is looking at release of lactate into the media in these neurons. And these neurons are actually quite old, so these are 30-day-old neurons. So these are just mature in a dish, and they release lactate. So this is sort of a control level of lactate. These first gray bars are what the LBSLs produce in terms of lactate, which is just sort of a measure of mitochondrial stress. And then with the ASO at a low or a higher dose, we can decrease the amount of lactate. So this is sort of assuming that we can decrease some of that mitochondrial burden. Next slide. Okay, so one of the slides, and I don't have it included here, but one of the slides that Manu had in his talk yesterday was looking at neuronal complexity, so sort of similar to what I had with the AAV9. We treated these cells and looked at them in a high content screener just to see how these cells develop in terms of neurite outgrowth, in terms of branching, and we can see that we get a dose-dependent increase in the branching and the complexity of these cells with ASO treatment over time. And that's an important figure, and I can pull that up if anybody wants to see it. So just to close out sort of the talk on the gene-targeted therapies, just, a consideration of administration and how this would work in patients. So there's a number of ASOs that are FDA approved, and you can see them listed here. So this is another figure from Manu's paper. And then, what we were hoping for, of course, is for spinal cord delivery. Our goal is to sort of bathe the spinal cord in these ASOs and the AAV9, and so this would require intrathecal delivery. And then so just, things to consider with an AAV9. This is a one-time bolus dose that you would hope has a long-term effect. An AAV9, an ASO, similar to nusinersen, requires repeated therapy since that affects transcription and because the ASOs are degraded. So this is just, things to sort of keep in mind when we think about gene-targeted therapies. Next slide. Okay, so just a quick update on some international research. So this is not me presenting on other people's data. I just want to talk about collaborative efforts. So go ahead to Finland. So Finland, this is a picture from the ARS 2023 meeting a couple weeks ago. So this is Dr. Henna Tinisma and Dr. Tina Rasila. So they are working on They're working on metabolomics and mitochondrial energetics for different RS2 disorders. And so they have patients' cells for RS2 and SARS2. And then I shared with them 3 DARS2 patient lines and their isogenic controls. And so what they're doing now is working on developing and differentiating these cells into neurons and doing metabolomics on them. So they've already completed metabolomics on the iPSCs, and the goal is to sort of work with us to see, what these changes might mean for LBSL and for DARS2, and then also to compare those to RS2 and SARS2 to see if we can find sort of some overlapping similarities in the function and dysfunction of these cells. So I've met with them online several times. This is the first time I met them in person, and it was really great to go over the data, and we have a pretty open understanding that, the only way that we're going to figure these things out is to sort of broaden our spectrum of researchers and expertise, and share cells and share information to get everywhere faster. So they're a really great team to work with. Next slide. Australia. So we continue to participate in Massimo's mission. Dr. Fine and I both spoke at their conference in April. They study DARS. So this is the cytosolic aspartic acid tRNA synthetase. So DARS2 we know functions only in the mitochondria. DARS functions in the cytosol. And what we found is that there's a lot of overlap in their data and our data in terms of what the mice look like, and then also in terms of how the cells function. So they have iPSCs and neurons that they have from DARS patients, and those cells look very similar to ours. And that gives us a little bit of hope. They're doing transcriptomics now. We just completed our transcriptomic study, so if we can find some overlap there, it might give us some information about maybe functioning of the functioning of the gene. That is not related to its translational duties. So maybe these genes do other things, and then we can figure out what those functions are and how to sort of repair that in the setting of LBSL or the DARS-related disease. Next slide. The Netherlands, so we continue to work with Mark Englund. Amina talked a little bit about that. When I was at the meeting in Canada for the R's, I met with Marissa Mendez, and she's a biochemist. She studies mitochondrial tRNA synthethases. A lot of what she does is looking at temperature sensitivity. So we know that a lot of LBSL patients don't handle heat very well, and it's a thing where these proteins actually destabilize in higher temperatures. And so she's done a lot of studies into that in other tRNA synthetases. And so what I'm going to do is I'm going to send her some of our cells. She can do some of these analyses, and we can try to understand a little bit of that functioning better. She also does amino acylation studies. So this is a very biochemical process that we're not really set up to do. And so she can look to see to what degree these enzymes are actually doing their job with the tRNA synthetases, with the tRNAs and the amino acids, with a little bit more finesse than what we can do. And then finally in the USA, so we shared our cells and our isogenic controls with Dr. Jose Abdenor at the Children's Hospital of Orange County. So his team will derive those into neurons and study mitochondrial function. So he does some metabolomics also, and he does seahorse assays looking at mitochondrial function. And he has a couple therapies that his team has tested on fibroblast lines, and so he's going to continue that research into our cells, and hopefully we can combine some data and some brain power to understand sort of those effects a little bit better. So I just want to tell you about 2 studies that we have coming up that I'm really excited about. So this is an NIH-funded study. So we wrote an R21 about LBSL, and R21 is an experimental grant, so it's sort of a high-risk, high-reward grant, and that was funded last summer, and we're really excited that we can get NIH funding talking about a rare disease. And so the idea of this grant is the thought that DARS2 does something else besides protein translation. And so what we said that we're going to do is we're going to take our cells and basically not freeze them, but we want to cross-link them. We want to bind their functioning in a dish. So we can do that by 2 different ways. We can treat them with a chemical that sort of locks proteins together. And then we can also zap them with UV, which actually binds the proteins to any sort of DNA or an RNA. And then we can isolate DARS2 in either of these, and then figure out what RNAs were bound to DARS2 and figure out what other proteins were bound to our DARS2, with the assumption that things that interact with DARS2 mean something about the function of DARS2. And so this is a way It's called interactomics. So this is a way that we can understand DARS2 function better to figure out, what it's really doing in the cell, who it's hanging out with, and what that might mean about its function. And so this is a study that we're working with right now, and hopefully we'll have some really interesting data on that in the next year. Next. And then this just got funded maybe about a month ago, so this is really exciting. This is a grant from the Maryland Stem Cell Fund. So this is part of the TEDCO family, and they fund high-impact research on stem cells. And so this is a grant that I wrote along with Dr. Lena Smirnova. So she is a PhD at the Johns Hopkins School of Public Health, and her team has developed a method to attach a fluorescent label to oligodendrocytes. And so basically, anytime there's a myelin-producing cell or a white matter cell, those cells are going to glow red. And we can take these cells and we're going to take our DARS2 cells, we're going to sort of trick them into expressing this red tag on oligodendrocytes, and then we're going to grow organoids, and we're going to see how these white matter cells sort of myelinate the organoids, how these oligodendrocytes develop, how they mature, how they interact with other cells. We can do a lot of high-level imaging. We can do a lot of gene expression and sort of figure out, why demyelination might occur in patients with LBSL. So we can really finely observe these cell types within a complex cell structure as they grow. So this just started about a month ago. Last week, I met with her student who took some of our cells over to his lab, and he's going to start the CRISPR experiments to get that going. So hopefully we'll have some really nice data on this too. So that's it. Happy to take any questions, and I want to acknowledge, so this list is sort of, the the current people in the lab in bold, and then the other names are some students that have been in the lab in the last few years, but in the years since we've started this project in 2017, we have a huge team. So a lot of people have done a lot of hard work, and we've gotten a lot of people to, to have an interest in LBSL and to really take this work seriously and help us move forward. So and thank you to all of them. And then I'll take any questions, and then we can kind of continue.
Was there a reason why you chose each one for delivery compared to other? It's very quick.
For the mouse model?
Yes.
So those mice are a CAMK2 Cree knockout, so they only are lacking DARS2 in neurons in the basically hippocampal area and the cortex. So this was, an attempt to be sort of a proof of principle experiment where the neurons sort of in the forebrain are the ones that are lacking DARS2, so can we directly deliver the AAV9 to those cells? I didn't have a slide on it, but now we have 2 additional mouse models that we're working with. One is an OLIG2 model, so hopefully, we have wherever oligodendrocyte precursor cells are, we're going to, knock out DARS2 in those cells, and then we have another mouse model for advilin Cree. So these are sensory neurons of the dorsal root ganglia and the spinal cord lacking DARS2. So for those animals, we would take a different approach. I might do the ICV for the DARS or for the OLIG2, but for the advilin Cree, we would do intrathecal. So the idea is just to, most effectively treat the cells in the vicinity where they are, hoping that we can see a difference, and then kind of go from there. The other thing is that ICV is much easier than intrathecal right now, so we're going to start with what we can do, and once we perfect the intrathecal approach, then we can try that.
Thanks.
I actually have a quick question
Yes,
on
please.
behalf of the patients. I know it's premature, but how are you envisioning it will be delivered to actual humans if we get this to that next step?
So the idea is intrathecal. It would almost have to be.
And would that be with sedation, or how does that play out?
I'm sorry today. I was just thinking that it might be. So yeah. So and just I don't know if I might have missed this, but yeah, intrathecal is the diagram is showing. So that's if any kids have had a lumbar puncture or a spinal tap, that's what it looks like practically. It's much more complicated though from the from the administrator standpoint, but and that's typically done, with the nusinersen and SMA, and that's kind of ASO delivery intrathecally is becoming just a much more common process in rare disease, and so we're actually like kind of looking at our infrastructure right now between Kennedy Krieger and Hopkins to accomplish it, because the demand is increasing. Hopefully, I think by the time we would be ready for LBSL to undergo the treatment, the infrastructure should be there. But another thing that they're working on, because this is not comfortable, right, to do intrathecal delivery. It takes a while. Like you said, sedation is required. This would be especially a challenge in children. They've got smaller spinal cords, et cetera. So we're working also on trying to spread out the interval that this would need to be done. So I think the best estimates are about every 3 to 6 months, if that is helpful. Ideally, it's even longer than that, but I think the technology isn't quite there. But it's acknowledged as a need. Yeah.
And do you envision that families would be able to receive the treatment at any facility nearby that has the infrastructure? Or would it have to be, they'd have to come to Kennedy?
Yeah, and this is, again, I've participated in a scientific advisory board for this industry group in December, and it was like a think tank for neurologists at different locations. In that case, it was just in the U.S. to talk about hey, what have you seen? What do you have at your institution? Trying to identify, areas, even major academic medical centers within states that people could travel to actually accomplish the therapy. So all I can say is there's large-scale efforts towards this, because it's just becoming more common and a bigger need in the field in general. Yeah.
Wonderful. I think we're primed for a break, and we're going to do a tech check with our virtual folks. I know they're having some challenges, but we have some new participants in the room that didn't get to chat. So I encourage you to please stay and chat with our group while we're going virtual on the presentation, and then stay for our next session. Thank you. Thank you, Mike, and welcome to everybody in the room. Welcome to our families who are virtual. I'm so happy to be here. As Mike mentioned, I am the Director of Patient Engagement, which is a pretty big title and a pretty big position for a pretty small organization. As Beth says, we are small but mighty, and that's very much the case. I've been campaigning ULF. I'm like, where's your Director of Patient Engagement? We've got one. Where's yours? We need that. So as you may know, Cure LBSL used to be called A Cure for Ellie, and the McGinns secured an incredible grant from the Chan Zuckerberg Initiative, which allows the foundation building that allows me to come in and be a director of patient engagement and create resources and work with our clinicians and our scientists and our patient communities to figure out what the needs are and make sure that we can get them. And then we can share with other smaller leukodystrophies that may not have those resources. Once we've created the template, then we can share. So if you're out in the hallway and you see the LBSL emergency protocols, please take them for your other groups and transform them. Email me. I'll send you the templates, and you can modify them for other leukodystrophies or other rare diseases. That's our goal. Next slide, please. So many of you I met for the first time virtually at our LBSL conference. For those of you who don't know, it's held every other year in Baltimore at Kennedy Krieger Institute. I was virtual with COVID, but I was grateful to be on. And during that presentation, accidentally having me at home and on the chat allowed us to connect with our families in Brazil. And those families in Brazil are now one of our largest patient populations, one of the most active. I can blast out an email and have a study filled in 5 minutes if it's available to that community. They are ready, they are willing, they are active, they're engaged, they want to participate. We have Portuguese interpretation on this call, and unfortunately, due to some technical issues, we lost many of our families, but we're being recorded, and I hope that they see this, and I hope that they feel welcome and part of this community. So our next, I think this should pop up. There we go. Our next conference is date to be determined, but is scheduled for 2024. And we expect it'll be at Kennedy Krieger, although we're already trying to coordinate, making sure that we're not competing with ULF and other activities. So stay tuned for some details. Okay, next slide, please. Actually, Mike, this is your slide. So can I pass this over to you for a sec if you want to jump in and share?
So I threw this together between a panel and the bar So it's probably missing a few things. But this is just sort of the journey of our foundation. I don't think I'm going to dwell too long of it, but yeah, so I hit Pit of Despair 1, but I haven't really gotten into Pit of Despair 2. Just kind of highlighting some of the major milestones. Starting a foundation, we didn't even know what we wanted to do. We started raising money, and we were sending mitochondrial cocktail powders, to people in Crimea. We didn't really know where to go with it. But it was really when we got together with Kendi Krieger and Dr. Fatimi that he kind of gave us a North Star. He said, you get $40,000 together and we'll start something. What something is, we don't really know, but we'll get something moving. And I don't know if he was just throwing some parents a bone to let them feel fulfilled that they were, taking action or what, but it's blossomed, and I think it's turned into something that has had some perpetuity to it, right? COVID kind of hit us in the gut a little bit from a fundraising perspective, but we bounced back pretty strong from that, and we've actually picked up some really strong pledges to help us get from where we are now, some of the therapies you just saw in the previous session, to getting those, into the actual patients, and we've gotten some money to help us kind of drive and kickstart into that. But yeah, this was under the thought that maybe we'd have some other parent groups here that were looking like, how do I start a foundation? What would it look like? How would it transpire? And what would we get toward the end of it? But our evolution gets us to about this point. And I did mention at the beginning, we actually have started a company because once you get some therapies that show some evidence of actually working, you get to the really expensive part of this, right? Large animal model studies, those tox studies, those other things that go into actually getting to whether it be an N of some or an actual clinical trial, just the operations, the manufacturing, the actual drugs, the delivery, these are really expensive measures. And we felt that on top of the philanthropic piece, that a investment arm would be a really worthy and helpful element for us to look into. So any billionaires in the audience, I have cards, we're happy to talk. But no, just kind of wanted to walk through that journey of our group and where we are now. But yeah, that's about it. That's a cure for LE to cure LBSL. Yeah, and that transition, it's not always great to name change. You do lose your SEO on Google a little bit. You've lost a little bit of your punch as far as where you stand when people are searching for things. But Ellie is a girl's name. Our former site had a tiara on it, and there's really no, it's not a gender weighted disease. So we wanted to make sure that we were inclusive for the boys that are picking up this disease as well, and to make sure that we created something that they could see themselves in if they do go to our website. So now it's much more oriented around any patient that is diagnosed. But yeah, thank you. I'll hand it back to Melody.
Thank you. And I just want to take this opportunity to point out, as I will through all of this, as I'm capturing the patient voice and the family voice and the caregiver voice, I'm just so impressed and struck by the family burden. Now, you go back to that slide for 1 second, Mike, if I just want to show, you have a family whose life got shattered with this incredible diagnosis. And in addition to your full-time job to have health insurance and Beth's job, and you have a child with LBSL, and you have an unaffected child who has their own needs, and I know we've spoken as well, and other people that I had the privilege to speak with, you're trying to do everything, and you're starting a foundation, and you're starting an investment corporation, and you've got all of this while trying to care for your children and trying to navigate insurance and trying to get appointments at the one time you're available and navigating these hospital systems. And so just for those in the audience who are on the other end of dealing with families with these rare diseases, just remember that the moment that they step in your door, they have already been carrying the weight of the world. And so just thank you for all of your grace and your time and your patience and your understanding with their questions or with their concerns or their fears. It's really, it's a lot. It's a lot. And I'm just so grateful to the community for being so open and sharing this information so that I can then be your advocate at meetings or in other opportunities. So. And I'm not going to call on the parents in the room, but please speak up. I don't want to put you on the spot, but I know this is your journey as well. So, I wanted to point. Oh, could you go back one to the website slide for a second? I did want to point out that the Cure LBSL website is up and running. We transitioned fully from a Cure for LE to Cure LBSL, and we have lots of resources on the website. So information about the foundation, but also patient resources and professional resources. And so for the professionals in the room, if you haven't had a chance to look, we've got lots of information about DARS2 and lots of information about the various research studies. And we also have the patient protocols that that Dr. Fine alluded to before, that available up there. And for the patient families, we have resources available for you. We've also gone global with our CZI money to try to capture. There's no other organization. There's no European equivalent of Cure LBSL. We are the international LBSL organization, and so we're slowly putting up resources in the languages that we've either paid to have translated, which is not inexpensive, and we've got our interpreters on. They know, it's a really big deal to have these materials translated and to have these meetings interpreted. And then we have, for example, Lithuanian. We have a researcher in Lithuania who was able to translate some of our materials for us as a gift to the foundation. So if there's anybody in this room who would like to translate any of our materials, we will happily accept your gift and put it right up there on the website. Next slide, please. So after the last conference, there were a few really critical issues that the families said they needed. They needed the support, the community, which is what the conference brought. They got the information, which is what the conference brought. And they tasked me in the next year between conferences to come up with other ways to improve their lives. And so one of the things that we did was our school advocacy. So if you go to curelbsl.org under resources school, or for the professionals, there's also up there under the professionals hashtag school. If you can go to the next slide, Mike. We have school plans, so these PowerPoint or PDF slides, and it has information in lay terms for teachers and administrators and disability coordinators to be able to help children thrive in school, and some highlights of what those needs may be. And this is just the cover page, but making sure that they're aware of mobility and balance issues, making sure that they're helping to conserve or not drain the energy of these kids so that they can function not only in school, but when they get home. Reducing infection risk so that we're not having those fevers that trigger the crises. Avoiding overheating and dehydration and fasting during the school day, and critically, the head injury prevention to, again, prevent another crisis. So we've got that information for thriving at school, and we worked with a special education teacher to help us. The other document is called School Needs and Accommodations, and it is a line-by-line checklist of various accommodations that could be provided to kids with LBSL or related leukodystrophies, very relevant to many of the other rare diseases. So please, please take it and modify it as needed for others. Sometimes you don't even know what to ask for. You don't know what's available. You don't know unless you've spoken to another family or you've seen a list like this. So that was our goal. Next slide, please. This is one of the highlights of my career. I love this. So many of you know my background's in public health and bioethics, and I had more fun and more professional satisfaction working with Dr. Fine and some of our other clinicians to create these protocols, because I can imagine how incredibly valuable they are. So what we did is, this is page 1 and page 2. It's we have them printed out downstairs if anybody'd like to see them. It is a 1-page document. Fold it up and put it in your backpack. Scan it, upload it, whatever. A place to put information about your diagnosis, your clinicians, your contact information, and then it has a quick summary. What is LBSL? Because the average urgent care doctor does not know what LBSL is. I'm not sure that the average pediatrician knows what LBSL is. I don't even know if the average neurologist knows what LBSL is. Honestly, like we're so spoiled.
Yeah, I can share on that one. This is extremely valuable because with LBSL, as a parent, the expectation that your pediatrician's going to know a whole lot more about it or how to treat it than you do is, it's not even a realistic ask. If they even, you would know better than I would, Dr. Fine, but I know they cover leukodystrophies. I don't know that they cover all the leukodystrophies as you go through medical school and cover each and every piece of it. But it's a very complicated thing. It's rare. It's not something that they generally focus on. And our doctors, earmuffs, Dr. Fine, but doctors are taught to emote and demonstrate a command of the subject matter with patients, so that there's a confidence, right? I think that's an important piece of it. But once you can work with them to give them information like this, that's not a you don't know what you're talking about approach to care, but just gives them a primer and gives them resources to learn more and to be more involved. Every pediatrician we've ever talked to has just dove in head first once we were able to get some information across. They're even taking things that they learned from like the mitochondrial specialists and using it with other patients. That's just a bridge that's difficult for, I think, everybody. It's difficult for the parent to try to get to with their doctor. It's difficult for the doctor to take in. But there are certain things that the doctors do need to hear about and know with this disease, and to fold them into the care that they're providing for your child on a more regular, non-neuro visit approach.
Please. And anybody in the room, please speak up.
Yeah, I mean, just kind of to add from the physician perspective, like, I was a pediatric resident at the Hopkins ER, which is like crazy and busy. And, we see a lot of kids with rare disease who show up with, a seizure or, some urgent issue. And it was such a help when some of the most organized parents would have a binder, with not always a face page. The face page is huge, right? Because then sometimes, I think on the other hand, you could potentially be loaded with too much information. We're not going to go through 5 pages, but having a succinct cover sheet so that, the new doctor is not completely overwhelmed, because that new doctor in the ER is the advocate for the patient getting the treatment that they need, and kind of like making sure the timeline is on point. The attending might have more information, but usually these training doctors are the ones who we're going to interface with first in an academic medical center. So if I can just tell you through my training, something like this, especially fitting on a page, would have been huge in my confidence and ability to take care of the patient. Yep.
That's fantastic. Thank you. And one of the things that I also love about this is not only are we sharing about LBSL, but we're also helping the ER or urgent care physician know how to prevent another visit, right? We're saying, please don't let this child get an infection in your care. Isolate them so that they're not back here in a week with a fever and worse symptoms. Please don't let them get dehydrated. Please don't have them fasting while you're waiting for the MRI. Please be aware that you could harm them beyond how they were already harmed when they came into you. And I think that's really critical too. And it's, in talking to Beth, just to not be the crazy mother, right? You walk in and you're like, I have some information I need to tell you about this condition. It sets you up for this weird dynamic. I've been there with my own children. And to be able to just hand something off and take it all off, and to have those professional resources. So if the doctor, we didn't want to take up the space on this page by listing all the references, but this is a well-referenced, curated document, I think, with many specialists, including emergency room physicians. And so we have the references online if they want more information, but we also just have this one page and things to look for. I can go to the next page too. So that was our cover sheet. You walk in with your page 1. Page 2 is your head injury protocol. And this is something that we want you to bring to school. We want you to bring to camp. We want you to bring to a child care provider with specific instructions for what to do with a head injury, because it's different. For example, in talking to the emergency room physician, her threshold for imaging with a minor head injury is going to be very different than your threshold for imaging for a person with a known leukodystrophy like LBSL. And so just to be able to give her the confidence to order it a little bit earlier than maybe her colleagues would have, and perhaps not have to fight with insurance, right? To say, we ordered it because in this condition, and we had the backup to do that and to cover it. I think that's really great. Our next one, when all of the grant cycles and meetings and FDA and everything is all settled down, the next one that I'm hoping that we're going to be working on is breathing and emergency response at home. What signs are concerning? What signs are to be expected? When do you act? How should you be prepared? What do you do? I know there's also, yes, Mike.
Yeah, again, just one important thing to keep in mind too is when it comes to anesthesiology, if they need to be sedated, or and then another big one is antibiotics. There's certain types of antibiotics that are not helpful from a mitochondrial perspective. So again, an ER visit, they might not know those things. And again, you've got some person with a business degree from a Big Ten school coming in that's saying, no, it needs to be a cephalosporin. They're not that interested in your opinion, but if you've got the sheets with you, then it's a little easier to kind of guide that conversation.
Thank you. And I know it can be a little bit hard to see up on the screen, but one of them, there's pharmaceutical considerations, and it talks about contraindications and additional lab work and which IV fluids to use. We also have references to an umbrella organization that covers the mitochondrial aspect, which just complicates things even more. We're saying many neurologists don't even know about LBSL, and then you've got the combination of it being a leukodystrophy and a mitochondrial disorder. And so to be able to consolidate that information into one document and share those resources so that the professions, perhaps they don't read the same journals, but they may want to get more information, as you had said. Lots of people are interested. So we have information up there, and we've got the references for who created the documents and where to get more information. Next slide, please. So let's talk about patient registries. That was another part that was really important. All of the patient advocacy groups are told to start a patient registry. But what they don't tell you is, one, how complicated it is, and two, how difficult it is to get the data from a patient registry into a form that's actually helpful. It's not going to be published in a peer-reviewed journal if it's not collected correctly. It's not going before the FDA as supporting evidence if it's not collected in a way that is acceptable to the recipient. And so adding to that challenge, we also have an international population, and so making sure that it's language appropriate and collecting the correct information. So what we wound up doing is we set up the International LBSL Contact Registry in 22 languages. So we're capturing people where they are in our database and they've indicated what they're interested in learning more about. Do they want educational information? Do they want to be part of research? Do they want to, be more, whatever it is that they're looking for? Do they want more resources? And we have a way to contact them. Then when we find out about research studies like the exercise study at Kennedy Krieger, and we find out who's eligible for participation, then we can reach out to those participants and invite them directly. We don't share the contact information. Even with our wonderful partners, we can't give the information, but we can request that information be sent to them, or that the participant reach out to them, or that we can request permission to share. Another wonderful update since the last time we saw everybody is that I don't know if we should call them a sister organization or a pseudo umbrella. They're the Cure Ours organization, which covers all of those. I mean, we're talking about the other related disorders. They got a grant from NORD to start up an I Am Rare patient registry. And it was one of the ones we had been considering just specifically for LBSL, and now it's going to be a much larger project, and it will include LBSL, but we're not running it, which is lovely. And, so we can put our funding to something else, perhaps to translations, to make sure that more people are included. In addition to that, I've been working really hard, including at this meeting, working with the GLIA folks. They're centered out of CHOP, but also Mass General and Kennedy Krieger, and it is the Global Leukodystrophy Initiative. They have a very large protocol called the MDBP, and we have been talking to them about not recreating the wheel and just enrolling more LBSL patients in these research studies and in their registries and in their biobanks, so that we're not all collecting little bits of data, but it's all going to the same place, and then having data sharing instead of little silos of data collection. So that's the current status of patient registries. If there's other opportunities, of course, we'll let you know. And if there's other ideas, please let us know. If anybody in the audience has other registries or projects that you think would be relevant to this community, we would love to hear it. Okay, next slide, please. Okay. My favorite part of the job. Actually, everything's my favorite part of the job. This is just an incredible, incredible, professionally and personally satisfying opportunity. But I have been tasked with capturing that patient voice, talking to people on Zoom, talking to people in person, talking to people at conferences, making sure that we get the LBSL voice. And critically, we had those, collecting those that patient voice so that we could share it with the FDA, which we did 3 weeks ago. Has it been 3 whole weeks? It's been a weird month. So about 3 weeks ago, we had you can go to the next slide, please. we had the opportunity to have a small closed-door Zoom meeting with the FDA. And when I say small, it was small on our end. We were restricted by how many people we could bring. So we were allowed to bring 7 families. We were allowed to bring 1 clinician. And as you know, we have a great relationship with many clinicians. So it's very hard to exclude. But we were allowed one clinician, and what, they gave you 5 minutes, right? 5 minutes, tell us everything you can about LBSL in 5 minutes, because they really wanted the meeting to be about the patient voice. So we provided lots of background information, and basically, this is a summary from my report, which is now 31 pages and still in draft. It was quite a meeting, and the way that we shared the patient voice, one was in a video where we had, gosh, I forget now, 18 participants from all over the world sent us video clips of what it feels like to live with LBSL. Many of you are on the Zoom call right now, and we had a video creative named Britt Faulkner who was able to put them together, set it to music, and make a really impactful video. Not to put you on the spot, but it was pretty special, wasn't it? It was really, I got to view the first draft of it at a conference up in Philadelphia, and I was sitting in my hotel room alone bawling. I thought it was really touching and beautiful. We also had Dr. Fine presented the clinical summary of LBSL, and I don't know how she did it in 5 minutes, but we captured 200-something voices and patient experiences. It's almost the entire known LBSL community in that little 5 minutes, plus our handouts. So that was pretty spectacular. We also put together an illustration, which I'm going to share with this room, of what it feels like to live with LBSL. We worked with artists right here in the Chicago area, a group called Ink Factory, and we were able to put together what is a poster, but for a presentation, it's actually a building PowerPoint illustration. So I think it's really powerful. And Dr. Fine and I are first I'm going to show it to you, and then we're going to go through it together, because some of the symptoms can actually be addressed. And so we'll go through it together and come up with ideas for how to address some of those symptoms. And then, of course, the critical part of the meeting was the patient story. So as I said, we had 7.5 families, because we had 7 families live on the call, and then we had a little video of a little boy with LBSL and his 2 siblings, probably 3 and 7 maybe, talking about their wishes for their brother, how they wished he could come to the playground, how they wished he could go to school with them, how they wish he could talk and walk and be like them, and how incredibly moving I just got chills, how incredibly moving it was to hear the voice of the sibling in addition to the voice of the parent and the voice of the patient. And then, as I mentioned, I'm working on the Voice of the Patient report, which will be linked to the FDA's website. And so it's becoming a permanent resource for them. Anything that comes across the FDA that mentions LBSL, they now have a little dossier on us, right? They're going to have all those handouts. They're going to have the meeting minutes that they took, and they're going to have the Voice of the Patient report that we create. So I'm really, really pleased with moving that forward. And next steps are PFDD, that's the patient-focused drug development meeting. That's already in planning at the low level. We're going to be requesting that so that we make sure that they have all the information, and that will be a much larger public meeting, so that they have all the information to move some therapies forward as we get to that point. Next slide, please. So this is the visual. If I show you, it's a building slide. So what does it feel like to live with LBSL? And if you advance it, it does have pre-recording on it. So I think if you just hit, actually, I'm going to step out, or you can advance it each. Is it not recording? Shoot, it should have. It should be auto playing. It may not be taking the audio from that computer. So it's losing a little bit of its impact as we don't have the narration. But basically, I consolidated the individual patient experiences that people shared with me, and we put it in this build-out slide. And so we went one by one by the symptoms. What does it feel like? So Mike, if I could have you back up, I'll try to narrate it just briefly to make sure we don't miss anything. So we started with the kind of obvious signs. If you met somebody with LBSL, what would you see? You would see that they have mobility issues. You would see that they have balance issues. You would see, maybe that they're holding onto a chair, or holding onto a person, or holding onto a service dog. Maybe you see that they're in a wheelchair. The next one. and you might see that that they're more prone to falling or that they have some fine motor difficulties. You may or may not see some of the neurological signs, especially we talk about the group 1 and group 2. Some of our families or some of our patients experience seizures or headaches. Some of them who are more mobile experience, as it was described, noodle legs, like they just feel like their legs are out of their control, or they may have pain or feeling of coldness. The feeling of coldness, Mike, you were the first one who mentioned it to me, and it's one of those themes that wasn't in the literature, but the families kept mentioning it. Ice cold feet. Do you have ice cold feet in your family? Ice cold feet, right? That's not in the literature, but the families were telling us that. Next slide, please. The heat intolerance, that's more of the mitochondrial part. So that's more, these may be more similar to the leukodystrophy community, but the heat intolerance and the loss of energy, that's more similar maybe to the mitochondrial community. And just that really strong feeling of being left out. Not being able to participate on the playground, not being able to go even, to a restaurant with friends. Some of our families were saying, like, if you can't find an accessible bathroom or you can't navigate, you're not positive that you can get there, you're left out and you're isolated. No energy. And this one, if you can see on the illustration, it's an agenda. One of our participants, who I think's on the Zoom call, was sharing what his day looks like. And the first 4 hours of his day are getting ready for his day, right? The getting out of bed and the ambulating and the stretches and the exercises and trying to transfer from a wheelchair to the toilet and transfer into a shower and bathing yourself. That's a half day of exertion just to get out the door. And then what? So just really highlighting that. Next slide, please. And just how exhausting the everyday tasks, the everyday daily living, just the act of getting dressed and out the door, how exhausting that is. And this is a big part, and unfortunately it skipped on the FDA meeting, so I'm really highlighting it in our report. We uncovered during these voice sessions some sleep issues that had not been documented in the literature. There are some generic leukodystrophy papers talking about gaps in knowledge, and sleep is listed as a gap in knowledge, but the actual sleep issues that the LBSL families are experiencing are not well documented in the literature. And so, it was really, I think I was four of these patient interviews in when I texted Beth, and by like 6 interviews in, I was reaching out to Dr. Fine and being like, hey, something's happening. And then we reached out to our partners at CHOP, Children's Hospital of Philadelphia, our GLIA partners, and said, are you guys checking for sleep? Is that part of your research protocol? And it's not. And so it's going to be because of this conversation. And so that's why this is so valuable. We're guiding clinical care, we're guiding research, and potentially guiding some FDA endpoints, right? If this is something that is impacting life and it's something that potentially is correctable, that may change a measurement. And when I say sleep issues too, they're not super well defined, they're just super common. Sleepwalking, night terrors, insomnia, non-restorative sleep. It's across the board. Everybody's mentioning some disturbed or non-restorative sleep on some level. So it's really interesting. The other part is talking about how hard people have to work to stay healthy and to stay strong. You've heard references to those mitochondrial cocktails. Everybody's on their own. I don't think are there 2 patients taking the same thing? I don't even know. Everybody is on their own, trying to research it, trying to get it funded, trying to get it made at a compound pharmacy, trying to, I mean, Mike, you could speak much more to that as your, as a dad, but it's not a small thing. And trying to not get sick, trying not, to not hit your head, trying not to, trying to live your life and stay strong and healthy, and how much effort it is to go to physical therapy or to do those daily exercises when you're exhausted, or when you're demoralized, whatever the issue is. It's not easy to stay healthy. So of course, and with that comes the mental health challenges. Whether it be anxiety or depression, fear is a huge one. In the families, we're hearing guilt and shame and other issues related to rare disease parents. It's not unique to LBSL, but it's highlighted in all of the communities, especially with a genetic disorder, that feeling of, it's unfounded, but it's common of the genetic component and feeling upset about passing that along. So we're now working with some genetic counselors to how best to help families process some of that information. Okay, next slide, please. And so this was our highlight slide right here. Wanted to remind the FDA that the damage is irreversible. This community cannot afford to wait, and they need a safe and effective treatment. And so we're working as hard as we can. All of those fundraising dollars. Before I came on as director of patient engagement, I was a donor to Ellie, Cure for Ellie. I was happy to donate. I had no idea where my money was going because I was just like, I trust them to do something. And then I came in on this side and I was like, oh my gosh, I cannot believe how much you have done. And I've told everybody in our social circle, you have no idea how much a motivated small community can do with every dollar that you do. And they sent it to the right people and used it so wisely and have used it's just incredible. And so just a reminder that this community doesn't have time to wait, and we're not willing to wait. They want, let's go. Let's everybody just keep going. And so the highlight message, which may be covered by these captions if you get to the last slide, is time. You can't see it, but it says, time is myelin. Don't wait every second. And so I feel that. There are days where I'm like, oh, my job allows me to have federal holidays off, but I got a job to do, and I can't wait. Ellie can't wait for me to, whatever. I'll take that day off next week. So it's not, we can't afford to wait. And so I'm just so grateful for the families for sharing their information. Mike, if you want to jump to the next slide. The other part that I'm incredibly grateful for is our response and our relationship. One of the things, as I've learned from working with other communities and other rare disease communities, is some of them are still looking for their researchers. They're still looking for their clinicians to be able to refer. And we've got this incredible network of collaborators that we can literally text. I can send a text or an email and know it will be read and responded to, as Mike and Beth can as well. We have these, as I mentioned, I reach out to Glia and say, are you guys doing sleep? And they said no, and now they are. Right? That collaboration that we identified a problem, we can get it to the right people, and they're acting upon it. So I'm very, very grateful for that. And we hope that translates to industry too. The one thing that I keep hearing, and I feel like it's my mission, is to de-risk this community, to make it so easy for industry or academia to come in and say, you have done everything you need to do to get this community ready. And as you mentioned Dr. Fine mentioned in the FDA meeting, this community is ready. They are research ready. The foundation is research ready. We're financed. We're go. Let's go. And so I'm hoping that if we de-risk it and make it so easy, we've got your patients, we've got your data, we've got your science, we've got your cell lines, we've got everything you need, that we can then focus it on the patient voice and get it done. Next slide, please. So one of the things that was amazing is, we had identified these two fantastic speakers for our presentation. And when I reached out and said, can you do a clinical update? Can you do a scientific update? Dr. Fine had the great idea to go back to this patient voice slide and go through it. And really highlight right now for the people in this room and the people on this call and the people who will be watching this video in the future, what can you do right now to address some of these issues and what's maybe on the horizon? So if I can maybe share, let's share the room. I'll give you the mic, and we can either go slide by slide again, or
yeah.
Or do you
And
want to just?
Well, I guess first, it's hard to know who's on the call, for Zoom. And I guess if there's anything, certainly if there's any comments about this slide and, identifying strongly with, any of these symptoms or the illustrations up here, please feel free to share. If it looks like there's something that's a priority for you that's not there, let us know. I think that this was helpful for me to see, the visual is also, I think, kind of really potent because, when we started the natural history study, you try to capture a lot of information, right? You don't want to also overwhelm. I mean, they're long visits. You don't want to overwhelm people with, fill out, 10 surveys every time, right? And it takes about an hour just to go through the history and to do the wearables. But I do want to make sure that we're open enough to evolving to include things that patients identify as priorities. So I think the sleep issue, that's a big, you've already touched on that is something that we had not been asking routinely about. So, and that can be easily addressed. There are surveys out there specific to leukodystrophy patients or patients with neurologic disorders. Actually, there's a resource called the NIH Toolbox, which I've used in other, in other areas, which is huge, short forms, really specific questions to kind of like focus in on the issues. And those are available remotely and they're free. So it's like I can kind of already imagine how we might try to like capture more specific information about that. So sleep is one thing that I see. It sounds like Leah's also recognized it. The fatigue, I feel like it comes up. We discuss it, but I don't. Especially it comes up in clinic visits. And that's another thing. I feel like a survey to capture that more specifically is helpful. Neuropathic pain, like that's not something that I have tended to talk to LBSL patients about. I see other patients with myelopathy, and that is a common thing, especially for older patients. So I feel like being able to kind of see these really helps to pull out some pieces of data that we're not gathering that we could actually easily start incorporating. And I think it's huge. So for instance, the sleep, the fatigue, the neuropathic pain, these are several categories of symptoms that I think are important because they could cut across to different groups with LBSL, right? So people who are no longer walking or haven't achieved the ability to walk, sleep is still critical. The fatigue is still critical, that they can still experience pain. And so part of what I want to do is make sure we're being inclusive for the whole community of LBSL, for these different age groups of onset.
Yes.
We're getting the information with the wearables, but not everybody is walking. Some people have lost the ability. Some will never achieve it. And so just kind of really broadening the scope to like hone in on what's important. And these things do turn into endpoints and outcomes for the FDA and for these trials. So, all that to say, I feel like it's nice how these things are sort of coming together organically. Yeah.
I agree. And I think one of the great things is, from my perspective, I'm getting more of the qualitative data that may not necessarily be published or may not necessarily wind up somewhere, but it will spark the type of research that will then be accepted by FDA. So I guess the one thing that I would say to all the clinicians in the room is, please use your surveys that are validated and that will, have meaning in peer review publications and have meaning to the FDA for future evaluation. But please always have an open-ended question, because I cannot tell you how many people do not fit cleanly into any of the validated measures. So if you could have that open-ended question where, is there anything else you want to tell me? Is there anything else how it impacts? And actually, Morgan, you got? Yeah.
No, one thing that I would love to have seen also is more nutrition guidance. Like, we emphasize the Mito cocktail so much, but every doctor, like Madison's weight has always been a struggle, and she's always hungry, always. It's very hard for us to kind of keep her weight in check, but nobody can give me any sort of clear, our pediatrician said, well, eliminate whole milk. Well, I don't think we've gained all this weight because we were drinking a quarter cup of whole milk every day. They say, just give her lots of carrots, but something more nutritionally. I would love to see how we could, if we aren't fasting, then we're still hungry, but then we're consuming more calories than we're expending because she's not that active. And so it's like a forever struggle for us. And I am so tired of getting sat down by doctors to be like, hey, we need to talk about her weight. And I'm like, well, she has no energy. You need to get her moving more. Yes, I would love to get her moving more. But anyway, sorry. That was just the thing that I don't see nutrition per se up there.
The slide's interesting too, in that these things are interconnected. There's a cyclical component to what's going on here, right? So, and the nutrition, I think, is a very variable element too, depending on where your symptoms are at, right? So, Ellie, for the longest time, was the exact opposite. She wouldn't eat. What we learned, we think this is correct, was that when she fasted overnight, when you get so hungry, nothing sounds good? That's basically what happened to her every day. So she just wouldn't eat breakfast. And she would go until the point where she was basically starved. And that's terrible for somebody with a mitochondrial disease. And the advice we got, good or bad, I'm not sure which, was stop worrying about a whole food diet or Mediterranean or anything like that. Calories. If she's just eating hot dogs, she's eating hot dogs. Just get food in her. And now we're at a different place. She's healthier in that regard. But with the slide, like you see the orthotics in the lower left corner. Orthotics have weight to them. The shoes that you're going to fit your foot into with an orthotic on are heavy. So now you've got weight added to your feet, and you're expecting someone to go do PT or get exercise. And then what happens if they don't get the exercise? Well, they lose strength. So it's harder to go try and make those steps happen again. And you've got this sort of downward cycle that is really unhelpful. And the nutrition's completely tied into it. But the trick here, and where I think it goes to, the clinic, is how do we invert that, right? How do we get them to a place where they can do the exercises necessary to be strong enough to carry the orthotic in the shoe and carry all the things that they need to do to live through their day, but then also do the things that they need to do to treat themselves to be strong enough to do it the next day. It's a difficult cycle.
And just to add on to that too, with the cycle of nutrition and GI tract, and it's mentioned up here on the slide too, but constipation and GI reflux is another thing that we've noticed. We don't have so much GI reflux, but definitely constipation, taking Miralax on a daily basis to mitigate that.
Yeah, I had, sorry. Yeah, that's another thing. Now, I've been actually now like more routinely gathering information about that with the visits, and like we have documented everybody on Miralax, and that makes sense. Like again, for older populations of patients with leukodystrophies that affect the spinal cord, it's like it's a big issue. Sometimes like with many, many years of disease, even incontinence issues. So that's something we're tracking. And one other thing I didn't mention is like practically what do we do with this information, like beyond it being like theoretical endpoints. I think an immediate next step with our partners in Amsterdam is standards of care guideline. And we don't have a published standard of treatment or standard of care in LBSL, right? Because we're getting a really good and precise picture now of the symptoms, right? From publications, from things that have yet to be published. And we have symptomatic treatments for a lot of this. We have experience from other leukodystrophies. So Mark England, who runs a natural history study in Amsterdam, had just, I think, a year and a half ago or so, put together a beautiful standards of care paper in ALD. So he kind of has a template. He's been doing it recently. He even expressed at our GLIA work group that was U.S. and international docs that he would like to do that. So I feel like that's definitely an immediate next step that can come out of this information to address things like, okay, what is that guidance on nutrition, right? So it's not a one-off. People have like a singular resource they can use. Yep.
That's fantastic, and I'm so grateful. If you look on our website, we have under the patient resources, we have generic leukodystrophy guidelines and handbooks, and they're wonderful, but this isn't a generic leukodystrophy, right? So you can pick and choose what you want, and we have some, pick and choose some resources from the mitochondrial community, but there isn't an LBSL specific, and there isn't another leukodystrophy like us that's close enough to say just use theirs. So we do have to create all of these materials, and we just have this wonderful relationship where we can work together to do it. But to your point as well, one of the other things that Cure LBSL can do and has started to do is start thinking about putting together educational seminars or meet the experts. And so, maybe our next one is have a nutritionist. And I know we already exchanged information to have a sleep specialist who can come and talk about the sleep issues specifically related to children with neurological disorders, even if not specifically LBSL, just general knowledge, and then maybe fine tune some of the LBSL resources, but also with the nutrition. So, when we were talking about the mitochondrial cocktails with some of our community, different people are taking different things depending on where they are. And when I had a community meeting with our families in Brazil, whatever they're taking is causing a lot of reflux. And they already have reflux. And so how do you get a child, and it's spread out multiple times a day. It's not like you just take it once or, once a week or whatever. Multiple times a day they're taking these supplements, and then it's giving them reflux and heartburn, or it's exacerbating what they already were going to have. And then how do you eat when you don't feel well? Or how do you how do you digest your food when everything is feeling unsteady? And so what do you balance? Or if you vomit, do you take it again? Like just making sure we have those guidelines. How do you know what to take? Or should you, if you take this mitochondrial cocktail with a glass of milk, does it invalidate it the way that, they say, like, don't eat your steak dinner with a glass of milk, have it with orange juice, you'll get better, nutritional uptake. That information is not currently available to our community. We need to get that information so people have the knowledge of how to mix and make sure that one isn't, invalidating the other or causing more harm than good or not causing any good when it was supposed to. And it's how expensive, Mike, I can't even imagine, cumulatively how much you've spent on these supplements and over the years in the compound pharmacy. But if you're going to do it and your kids are going to take it and they're going to, go through it, you want it to work or at least not cause harm. So go to the next slide. I think we're actually going to wrap up. We had repeated this slideshow again at the end of the slide deck without the sound, but then it turned out the sound was on. But this was your last slide. If you'd like to address, and then let's just open the room for questions and discussion, and then we'll end in just a few moments.
And actually, we've basically sort of covered these things throughout, which is great. But the final thoughts are thinking about, as in other leukodystrophies, maybe more moving towards more of a formal classification of types, because sort of the issues and the primary symptoms and challenges do tend to be different based on age. At the end of the FDA patient listening session, it was asked, what's the one thing, like if you could address one thing with a cure or, with a treatment, what would that be? And I was sort of struck. First everybody was a little hesitant, but that differed, right? Based on, mostly age of onset of the child, right? Like, so I think one mom, their child really struggled with feeding and nutrition, right? I can say one family, I don't think they spoke up in that moment, but the respiratory issues, respiratory insufficiency ultimately led to their child's death, which is horrible. And that's not something that I hear or see a lot, but like that's critical. Then of course for others, it's, the ataxia and the walking issues. So all of that to say that it varies, and it's a rare disease, but we still kind of really need to think to be inclusive about all these groups. And I think we're in the process of doing that, and I'm glad that the FDA is hearing this, and we'll keep doing, we'll keep moving on that pathway with them. I think that's basically it. Yeah.
One of the things I'm very grateful for FDA is they're moving forward from our understanding in not only having research endpoints for the whole study, but also individualized endpoints. And I think that our patient listening session really highlighted that individual priorities or individual experiences need to be considered. I'm really struck by two of our families that I interviewed where, if I was quantifying on a rubric, I would say that their children are extremely severely affected in terms of, day-to-day experience. But what they both have is incredibly peaceful, happy, not, they're not irritable children. They're not, they're generally very content. And so, a mild side effect of irritability that FDA may tolerate may not be tolerable for that family because it would be a huge change in that child's quality of life to be irritated or to be upset, whereas somebody else might, if you have a teenager, you're like, how would I even measure irritability in a teenager? Like, it just is, right? That may be something that's acceptable. So, just really being able to make sure that everybody is just listening to the patients and listening to the parents for what is acceptable or not acceptable to them, I think is a really big take-home message for all of our clinicians, researchers, and regulators. That each one is going to have their own priorities. And so with that, I think we are officially wrapped up for our slides, and I would just love to take the last remaining minutes of this meeting to include all the members in our audience and maybe share. I know not everybody came in for the beginning, so maybe there's something we could cover that we already discussed, or is there any questions from our group, how we can include you in our discussion and bring you in as partners, because you can see we're a tight community with, and we take care of care of our clinicians and research, right? We love on our whole research community, so we would love to include all of you as part of that. May I pass the mic to anybody? Any questions? Jojo.
Not really a question, but I think with all the great work you guys are doing, and especially the educational material, with the different emerging gene therapies basically getting out there and becoming more and more frequent, I think it would be also helpful to add, potentially different needs after receiving any sort of gene therapy to the educational material. And the reason is because most medical schools don't teach anything about gene therapy, and the needs are often unknown prior to the trial. We discover a lot of needs that occur while patients receive these gene therapies in a trial. So a lot of that is actually not known. But I think adding that to the educational material might be helpful to make your next, hopefully not next, but your next emergency room visit a little bit, less anxious or anxiety provoking.
For that insight.
I have a simple question. I missed the early part of this session. That I just want to know, what is the proportion of a genetic cause for this type of disease? You mentioned that ASR2 and also ASR. What's the proportion of those two different causes?
So I guess the question's about the different, like the family of tRNA synthetase disorders,
Exactly.
and you're wondering about the prevalence, Yeah, so I think Christina talked a little bit about these, like there's,
I missed that.
for each, yeah, no worries. And so I kind of, just as a nod in case she wants to contribute as well, but, for each of these mitochondrial tRNA synthetase disorders, like they correspond to the different amino acids, right? And we're still learning about some of them, like just really a handful of cases that are known. But a lot of them do have neurologic symptoms, right? Maybe not even a neurologic, maybe not a leukodystrophy per se, but neurologic symptoms as well. So, I mean, to my knowledge, LBSL or, DARS2 disorder is the most prevalent one. So we know of over 200 cases that are published. You're asking about prevalence of the others?
Between the two, and also my understanding, this is a loss of function disease.
Well, yeah, maybe that's a little bit more complicated. I'll defer to you there, but oh my goodness, I think that ours too. We maybe know of it's maybe 30 cases right now,
Yeah.
and I think that's the second most prevalent one, right? So. Kind of as a group, like if you consider all of them, there's like a relatively large prevalence, but like LBSL was the first one described in 2003. I think ours was a few years after that. They're sort of like sequentially we're learning about them. So unfortunately, the prevalence like vastly decreases.
I see.
Yeah.
And also, it sounds like this is kind of a more concentrated to certain geographic or ethnic
Yes.
groups. So is there any grandfather effect or something?
Well, I mean, I can say just it is known there's a high carrier rate in Finland for some reason there's like 1 in 95.
Oh, that's pretty
Yeah,
high.
so we're actually like really happy to have Finland now participating in the natural history study to capture some of that.
Very, very
There's certainly European background predominance and sort of Eastern European as well.
200, that's quite decent.
Yeah.
Big number, actually.
And I actually have an update on that, which I didn't add as a slide because we haven't had our first official meeting.
Yeah.
But as part of the Chan Zuckerberg Initiative, we're part of the Rare as One network as grantees. And we have been invited to be and included in a Broad Institute prevalence study of the DARS2 variants. And so we have our first planning meeting in August. I'm really excited to update the community as soon as I have something to update. But they're on it now. We sent them all of our information, and they're going to help us estimate the prevalence of the DARS2 variants.
Okay.
And we know many many of our population are misdiagnosed right now, right? We have a lot of people who think they have MS, who are adults, and other conditions. I don't know if folks from the virtual group want to jump on, but as I was talking to people, getting the list of misdiagnoses before we got to LBSL, or before they found the DARS2 mutations, was high. And so, just going, we don't know what the true prevalence is, but based on the estimates from genetic testing, we're hoping that Broad can help us figure out what's brewing. So.
Thank you.
Anybody else? Let me see if there's any questions from our virtual folks. Please ask.
May I ask from the 200 that you know about, how much are adults in this?
I'll just speak loudly. I haven't actually looked at that exact breakdown, and we could. Is there a reason in particular you're curious?
I'm adult neurologist.
Yes,
so
fair enough.
I only know adult patients.
Yeah.
And I thought it was an adult disease. more. If you see only adults, then you think it's adult disease, but it's obviously it's a spectrum. So I want to know if this is starting always in childhood, or can it also start in adulthood?
It can, yeah. So, and that's, I think probably there's a paper from Mario van der Knap's group in 2014. I think Van Berg was the lead author. I think there were like 70 patients, and they're enrolled in that natural history study, and it does give sort of a nice breakdown of, at least, at that point, the onset, age onset, and sort of, I just can't remember those numbers exactly, but that's a good resource there. But yeah, there's this like basically antenatal onset, infantile onset. It can be, most often it's in childhood actually. So that's kind of the prevailing thought, and it's borne out in the U.S. Like most of our patients are pediatric actually in the study. We have a harder time recruiting adults, but on the other hand, I think probably because Maria van der Knap started studying this group like 20 years ago, most of who she has, that's still in contact for the natural history, they're adults now. So like that's one reason that for their IRB protocol, they're only recruiting 16 and older. So that's, I guess, an advantage of having these multiple sites is that we can have, maybe we have a site that's predominantly pediatric, a site that's predominantly adults. I know in Finland and Brazil, they're all comers. I think we're still understanding more about the prevalence in different groups, but sometimes symptoms like clearly don't show up until adolescence, or later.
Yeah, it's also important. If you're asking whom should I address for the information, for example, I mean, if it's occurring in adulthood, there may be other differential diagnosis, or you're asking people who are dealing with multiple sclerosis, for example, because it looks like multiple sclerosis in some cases, and so the information that you are providing should address this issue. So you need, for example, for the pediatricians, maybe other information than you need for adulthood neurologists. So
So the symptom severity decreases with the age of onset. So it's much more severe if you get it younger, and if your symptoms come on when you're an adult, they're much smaller.
In other diseases, leukodystrophy as well.
Yep.
And the patients that I know are very slow progressing, so it's a very benign disease, whatever, in adulthood.
We have a handful of people
but
that
maybe this information is only minor information because it's one center, and so these registries and the information worldwide is very important. But I want to say that the that it may look different in adulthood, like in childhood. So this take this into account.
Yeah, that's what we've seen too. Just so my wife's been sort of the magnet for patients. Very often, she's the first person they find that offers a different kind of voice than the medical community might. But we found that a lot of people who get it in adulthood, or their symptoms start in adulthood, they're so mild that they might not even attempt to find a medical solution to it. It might just be fatigue. Stairs are difficult, right? Things like that are that kind of minor that they might not even seek out an MRI to see what's going on. But again, it is a spectrum. We're saying there's the types, and I think there's some reality to that, but there's also potentially just a complete no type, but spectrum after a certain point. Anyway.
I actually have a question to send back to you as an adult neurologist. What would be the most effective way for our organization to reach the adult neurology population to say, hey, think about LBSL in some of your patients that, that may be presenting in this way or that? Would it be taking out an ad in a journal or attending a conference, or how best to get that on the radar of the adult neurology population, practitioners?
Well, in my experience, it's I think the MS persons are the right persons to address first, because this is an important differential diagnosis for adults with a chronic progressive myelopathic syndrome, which has not the typical features that you find in multiple sclerosis.
Yeah.
For example, oligoclonal bands, for example. And but on the other hand side, it's very impressive that you find the MRIs. They can look like a multiple sclerosis. So if you don't do the spinal cord, you would miss the diagnosis.
I was going to say this.
So it would be interesting to have some, let's say, one-sheeter red flag page, one pager for the information, giving some red flags in the differential diagnosis of multiple sclerosis and other spastic paraparesis conditions.
Yeah. I like that idea. And so just thinking about MS again, I mean, one, you're right, the spinal cord is so important, and sometimes they're just getting the brain. I mean, I feel like a lot of times there's reasons to get spinal cord imaging in MS patients anyway, but it's not always done. And hopefully you saw earlier, I had a couple slides where I showed the T2 changes in the spinal cord. They're throughout the whole cord, where it's not that sort of thumbprint, scattered lesions like you see in the MS. It's if they get the cord, that should be a tip off, like something's going on.
The spinal cord is really diagnostic.
Yeah,
There's virtually no other disease that looks like LBSL.
Looks like LBSL.
We were told our diagnosis journey wasn't that bad, but we were initially told potentially spinal cancer, but then it was too smooth, the change in the spinal cord, and then tethered spine, right? So that, and then we have a bunch of patients who've, like kids who go off to MS camp in the summer, right? That's a pretty common one that they've been given. So yeah.
I've also heard some misinformation. So we've actually had a couple of really good diagnoses come up from the Hopkins MS clinic. A woman with RS2 who had been treated as primary progressive MS for 10 years, and then she was mute and quadriplegic, and basically had lost all function. And then it was like, well, maybe this is a genetic disorder or leukodystrophy. Also, in fairness, her MRI initially did look a little bit more classically like MS, but the lesions evolved. That's RS2, but I guess the point is, in some of the discussions with neuroimmunology fellows, adult neuroimmunology people, and the MS clinic, there's this misunderstanding that there's spinal cord involvement in leukodystrophies, which is to me crazy, but I'm super specialized, where some of these younger clinicians being trained are not thinking about getting the genetics or considering leukodystrophy diagnosis when there is spinal cord involvement. Again, that's surprising to me, but being super specialized and mostly focusing on leukodystrophies with myelopathy, it's. But yeah, I feel like your red flags idea is important, and maybe like kind of as we're putting together, thinking about standards of care and diagnosis, that is something that should be emphasized, because I do really worry about, just in general, about adult presenting patients getting genetic diagnoses.
I think that's an interesting thought, though. It's an interesting thought to maybe try and insert ourselves at like an MS doctors scientific conference to say, yeah, look at this. This looks similar, right? But it's not the same thing.
That's what I think we were just talking about for general at the ULF patient advocacy meeting last night, talking about having a QR code and a table at conferences. Thank you. Just to get doctors thinking about leukodystrophies in general, like what are some of the red flags if they're not thinking leukodystrophy. But I think this is a really targeted area to get to the adult population and encourage, come up with those red flags and encourage, the appropriate next steps, or, how often are adults being sent to get genetic testing versus, I know the peds much more on their radar to be thinking genetic as it plays out.
The adults don't have as many of the cognitive issues as well. So like that's another way that, in terms of the diagnosis pipeline. So I had mentioned most of our young LBSL patients have gross fine motor speech delays, this learning issues. So that threshold is higher. The adults that I know of in our study, they're lawyers and professionals, and didn't need extra school supports by and large. So yeah.
Dr. Bertini, anything you'd like to add? And you've done some amazing work. We're just having just a general discussion. We'd love to hear what you're up to, or some future paths for advocacy or for research or study.
What I can say is that I understand that there's a lot of interesting news on this disorder, so I hope, the antisense oligonucleotide strategy is something that is working already in one disorder, and I think who follows SMA patients can confirm that it's now here, of course, we're related to a brain disorder, so there's something new. I mean, there's no experience on a brain disorder, so on one side, I suppose that the ones that are involved in the antisense oligonucleotide project can have the feeling, not yet perhaps, on which would be the magnitude of change that they can obtain with this strategy. Of course, gene therapy is something that seems to be more efficient, and I suppose they will try to cover the other patients with different variants. So I just want to recall that in that paper that Dr. Fine presented on the severe cases, we participated with some patients, and one of the category of patients that had infantile onset leukodystrophy in that series was asymptomatic, at least until the paper came out. So we don't know the story of that single patient. And that was a particularly concerned of that, because we're just following this little child with that seems to be so far not, improved a little bit, but then now is stable. Mm-hmm. And so also in that infantile onset forms, generally they have they recur. They have more rapid course. Mm-hmm. You can have an exception to that.
Yeah.
And they don't seem, these infantile leukodystrophy forms don't seem to have, at least so far, so they have to be followed, seem to have spinal cord involvement.
Yeah.
But we don't know. I mean, we have only a short follow-up with those patients. So
yeah.
The others are probably, we tried in that paper, to correlate to the yeast modeling and to see if there was some correlation with the severity of what we were observing. But probably yes, in most of the most severe cases, you had a truncating mutation in one allele, and the other ones were mostly missense. But of course, the genotype-phenotype correlation was not completely explainable in those cases. But
Yeah.
This is what.
And that's still the case, and we're, is, is, yeah, yeah.
But I mean, I understand the gene therapy, you know, it's a way to cover all the other patients that don't have the intronic mutation. So, I mean, the project is very interesting. I mean, so it's moving.
We have I've presented on a couple times, but we have a paper coming out hopefully within the next week or two. We did the transcriptomics on our patient lines, and it looks like depending on the type of mutation, if there's splice site mutations or if there's at least one missense mutation, that the gene expression profile of cells is different. So, you know, when we kind of got to that point, it became clear how complex this disease is and how complex some of these downstream effects are, and we thought that gene therapy would be sort of the best option to remedy some of these things. And, you know, we're still working to try to figure out mechanism and try to understand some of these, you know, effects of certain mutations or if there is a phenotype correlation. But we thought that, you know, it would take much more time than we had to understand it to that degree. And if gene therapies are progressing, that's sort of our best avenue.
I suppose that for the gene therapy program, you will have to go to the step on trying to address safety issues in the primates, like classically it's done. I mean,
Yeah, so
take some time.
these are sort of things that we're kind of working through now, how to navigate that road and how to involve other partners to do, you know, those are not things that we would do. Those are things that we would get others involved in. So definitely.